GM crops pose two types of potential risks: food and feed safety, environmental safety. Allergens, toxicity, anti-nutrients, and a change in nutritional characteristics are viewed as food and feed safety risks, while the following is seen as environmental risks: effects on biodiversity, modification of soil, insect resistance to Bt protein, weeds becoming resistant to herbicides, superweeds, crops becoming invasive, gene flow of the inserted genes to wild relatives or non-GM crops. How are risks assessed? Food and feed safety. Food safety assessment is carried to detect any hazard, nutrition, and concerns that may harm the health of consumers. The following are assessed: identity and source of the gene, composition of the new crop, effects of processing, cooking the GM food, stability of the inserted gene, the new protein produced by the inserted gene, the function, toxicity, allergenicity of the protein, any secondary effects from the new gene on bodily functions, any difference in composition of critical macro, micronutrients, anti-nutrients, endogenous toxicants, allergens, and physiologically active substances. Potential intake and dietary impact of the introduction of the GM food. These potential risks are identified through a number of scientific methods. Number 1. Molecular characterization. Molecular characterization of the inserted gene and new protein is carried out on the inserted gene and the protein produced by the gene. A number of analyses are carried out that provides data on understanding on where the gene is located and inserted, genes adjacent to the transgene, number of inserted genes, digestion of inserted genes in animals, stability of the gene and its trait in the crops, unintended consequences. Number 2. Feeding studies. Feeding studies are conducted using animals and they provide the following safety data. Substantial equivalence. Conventional crops are compared with the same GM varieties to see if there are changes in nutritional composition, toxicity, or allergenicity. The safety of the new crop for humans and animal. The safety of the product derived from animals raised on transgenic feed. Environmental safety. Environmental risks are assessed based on the potential hazards and the harm caused by these hazards. These are assessed in different stages. Contained use. This is the initial experiments in the laboratory, greenhouse, or any facilities that have no contact with external environment. The scientists involved must notify the regulatory authorities about their experiments. Hazards that are monitored are: A. The plant's capacity to survive, displace other plants. B. Will they cause harm to animals? C. Will they cause harm to microorganisms? D. Do they have altered interaction with plant pathogens? Confined trials. These are experiments in open field but with restricted access to human and animals. These experiments provide information on environmental interactions and agronomic performance of the GM crops. Field trials. These are open field trials just like a commercial farm but in a designated location. Areas evaluated are: weediness, susceptibility to pathogens, effects on non-target organisms, unintended effects on the target organisms such as resistance development. Can the inserted gene or sequence result in a change in management of the genetically modified crop plant that has a negative impact on the environment? Can the inserted gene or sequence cause adverse changes in biogeochemical processes such as changes in the nitrogen cycle? How are risks managed? Food and feed safety. Approvals are not given if there are adverse effects found at the development stage. Environmental risks. Gene flow to non-GM crops. Isolation distances are maintained, which is the minimum distance between GM and non-GM crop fields of the same species that should prevent a cross-pollination above threshold levels. To determine the isolation distance for each crop, population, size, distance, and flowering timing between donor and receiver fields, as well as local wind conditions, are studied during field trials. Crops can also be cultivated at different times to avoid gene flow. For example, farmers agree to coexist by planting different crops at one time. Conventional soy and GM corn are planted at the same time to avoid cross-pollination. Gene flow to wild varieties. GM crops that are capable of cross-pollinating valuable wild varieties are planted far away from where the wild plants are. Insects resistant. Farmers have refuge in the farm. Refuge is section of the farm where non-GM crops are grown. This allows insects that feed on Bt and non-Bt to interbreed, which slows down resistance among the next generation insects. Scientists insert more than one type of Bt gene, which is called stacking or pyramiding. This also slows down the development of insect resistance to the protein, as now more than one type of protein is produced by the crop. Weeds developing resistance to herbicides, stacking or pyramiding is done by scientists where more than one type of herbicide-tolerant gene is inserted. Farmers can now spray different types of herbicides in rotation, which slows the development of resistant. Irreparable damage to the environment, such as crops becoming extremely invasive. Approvals are not given if there are adverse effects that cannot be mitigated. Approvals may also be revoked if these damages are detected during post-approval monitoring.
For more information on biosafety in Sri Lanka, visit biosafetyclearinghouse.net.